Okay, this is key assessment 6C and we're on question 2 now. So this is about spermatogenesis and we've got a transverse section of a mouse seminiferous tubule. Poor mouse. So we're working, what's going on here, we've got a basement membrane, so that's sort of like the outside of the tubule. Then obviously we've got that sort of sequence of spermatogenesis, so going from spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, spermatozoon. So we need to kind of remember what's going on. So we've got mitosis and growth, so these two are diploid. Secondary spermatocyte, we've done meiosis 1, and that's haploid. Spermatid is the result of meiosis 2, and then we've got differentiation here. To make us a sperm. So, question then. Oh, and then we've got this Sertoli cell, and what I can remember, I like to call them nurse cells, so they do nourish and protect functions, and of course that's going all the way from the outside to the inside, so if you could sort of drew a little line around it, like that. Okay, identify cells in the section that match the descriptions listed. The cells can match more than one description or none. Oh, excellent. So, undergoing meiosis, so we need to remember that we've got the two divisions. So it's the primary leading to the secondary. So primary and then we've got the secondary ones doing meiosis 2 to form spermatids. Ooh, they've left you a lot of room there, haven't they? Spermatocytes will produce more cells by mitosis, so that will be spermatogonium. See now the value of annotation. Provides protection from the male immune system. Sertoli cell will undergo differentiation to produce a fully functional cell. Differentiation's there. Spermatid. Speeds all that process up if you do your thinking initially. This is a pretty standard question too. Explain why there'll be more spermatids than spermatogonia. So think about one spermatogonium. It's going to do mitosis, produce 2 cells, produce 4 cells. That is it. Each, and I could have put the word because in front, spermatogonium divides by the 2 meiotic divisions to produce 4 spermatids. Flipping over, and we've got a graph, and we've got another slide there. So, the Sertoli cell, which all we've done is really say, oh, it nourishes and protects. We've got some details here. A prominent nucleolus. What do I know about the nucleolus? It makes rRNA and ribosomes. And large numbers of ribosomes. What do I know about those? This is core, isn't it, again? That they do protein synthesis. Specifically translation. It is known that these, that's about Game of Thrones, that isn't it, that these cells are involved in providing nourishment to developing sperm cells. Suggest what nutrients Sertoli cells provide Give reasons. So we've got prominent nucleoli producing ribosomes, ribosomes producing proteins. I'm thinking the nutrient is proteins. Well, proteins. Why do I think that? Because the nucleolus produces ribosomes, and a large number of ribosomes produce protein. Okay, another function of Sertoli cells, who knew, is to remove debris, oh that doesn't sound very nice, from the lumen of the seminiferous tubule. Ooh, I don't want to think about that. Name the process. So we're removing stuff. You've got your seminiferous tubule, hole in the middle. You're removing bits and bobs from there into, I don't think about what those bits and bobs might be, but anyway, bits and bobs back into the Sertoli cell. How are they going to take them up? They're going to be quite big. I'm thinking debris suggests chunks. So, name the membrane transport mechanism that removes chunks. Endocytosis. Or if you're going to be more exact, you're looking at solid particles, so it'd be phagocytosis. Anyway, on to more pleasant topics. Menstrual cycle in cows. Miss, we've not learnt cows. It's controlled by the same hormones as women. Well, that's handy. The diagram shows the changes in the levels of some hormones of a cow over a 22-day period. So, the graphs you've done are this. You've got a big spike of FSH and LH. So LH, FSH. You've got then, it's changing colour. You've got estrogen, and you've got progesterone after ovulation. Everything dipping towards the end. That's humans. That's the graph that's in your head. Or not. So we've got a spiky arrangement here. This is the spiky in the middle. So whatever hormone that is, hormone A is going to be LH. Big spike causes ovulation. Prior to ovulation, we've got estrogen, so that's hormone B. And then post-ovulation, we get this hormone released, hormone C, progesterone. Not too complicated at all. Estimate the length of the menstrual cycle shown. So we're going from, where have we got a repeating pattern? So that's day 1, isn't it? That's day 19. That's where I would take it. Day 1, day 19, 19 take away 1, 18 days. So remember, when you're doing tying, you need to look at repeating patterns. Another good place to have done that might have been day 2, day 20. So it gives you peaks or your troughs. 20 take away 2, 18, super duper.